Hi, everyone. Welcome to today's coastal state discussion. I hope you all can hear me. If not, just give me a quick shout in the chat. But I'm Meredith Haas from Rhode Island Sea Grant, and I will be your host and moderator for today. Um, and for those of you that are new to the Coastal State series, it is a dedicated forum to highlighting ongoing research focused on marine and coastal issues that impact Rhode Island communities. And you can find more information about the series on Rhode Island Sea Grant's website, where you can find recordings from past events, such as um, last month, where we focused on the challenges and some of the opportunities that the multi-use framework presents for small coastal business owners. So if you weren't able to join us in November for that discussion, you can visit our website. You can also visit our YouTube channel where that recording is held, as well as other previous discussions on various topics. And so just to give you a brief overview of the format for today, our guest speakers for today will have a presentation that will be about an hour in total, followed by Q&A. Our speakers may ask for your participation during their talks. So we just ask that you use the chat and the Q&A option at the bottom of your screens to ask your questions, make your comments, and we will get around to addressing all of your thoughts and questions. And today's discussion will also be recorded. Like I said, we'll also be on Rhode Island Sea Grant's website and our YouTube channel. And so to kind of get this going, let me get out of the way. Let's get into our topic for today, looking at the potential for sea urchin farming in Rhode Island and in New England. And so what's interesting is that sea urchin roe, or uni, is a highly valued commodity across the world, estimating about $60 per pound. And, our speakers can correct me on that. But as wild stocks face overfishing and habitat loss, farm sea urchins may provide a sustainable means to meet market demands, as well as add value to existing shellfish and kelp farms by reducing maintenance costs associated with biofouling. And our speakers today are Dr. Coleen Suckling and Dana Morse. Dr. Suckling is joining us from the University of Rhode Island, where she is a marine ecophysiologist and associate professor of aquaculture and fisheries, where much of her research focuses on understanding how commercially and ecologically important organisms interact and respond to differing conditions. And the goal is to ensure future sustainability of seafood and ecosystems. And she brings 20 years of experience in sea urchin production, which includes running research hatcheries, integration of sea urchins with salmon and kelp, aquaculture to reduce environmental impacts of those operations, as well as developing formulated feeds for sea urchin production. And Dr. Suckling is currently leading projects looking at the feasibility of rearing purple sea urchins to diversify Rhode Island's aquaculture, as well as expanding sea urchin production in the Gulf of Maine and northern New England. And Dana Morse, who's joining us from Maine Sea Grant and the University of Maine today, is an aquaculture lead and senior extension manager for Maine Sea Grant. And he's been working with Dr. Suckling and others on this effort to expand green sea urchin aquaculture both within and outside of Maine. And so he's going to be elaborating for us today on some of those efforts. And so with Sorry, I muted myself. With that, I'd like to hand it now over to our guest speakers. Thank you, Dr. Suckling and Dana, for joining us today and sharing with us your research and all the initiatives that are going on in New England.
Great. Thank you so much for that wonderful introduction. So I'm just going to get my PowerPoint set up. So excuse me a second. Okay. Okay. All right. So hopefully you can see my slide there and you can hear me okay. And do we need to have my camera on? That's strange. Okay. All right. So so thank you for that wonderful introduction today and the opportunity to talk about these projects. We're very excited to be talking about all things urchin related. And And it's always a wonderful opportunity to be able to talk about sea urchins for a solid hour. My passion projects. I love working with sea urchins. They're one of the most wonderful creatures on the planet, but of course I'm very biased. So anyway, today we're going to be, so I'm going to be doing the role of like looking at sea urchins as a seafood product, because perhaps some of you may not have come across this before. And then I'm also going to be looking at the covering it as a fishery and looking at the emerging sector of aquaculture. And I'll be going through some of the general practices that we apply with this, and then we can use that to kind of help build on explaining what our recent efforts and research has been involved in, and talking about the relevance to Rhode Island. And then we've got the wonderful Dana Morse, who I'm excited has joined us today, all the way from sunny Maine. And Dana's going to be talking about aspects of the projects where he's been a really kind of important role. He's had this really important role of increasing awareness and uptake of green sea urchin aquaculture in New England. And then he's going to outline some of the wonderful informational resources that are open access and available to everyone. I believe Dana's going to be promoting some discussion during this today.
Indeed.
All right, so we're going to start with the star of the show. This beautiful specimen here is the green sea urchin, Strongylus trotus drobachiensis. This is the only current commercial species that we have here in New England. You can find these wonderful round spiny creatures living on the seabed. And we can find them mostly north of Cape Cod. They like to live in cold waters, so waters that are below 18 degrees Celsius. And they spend their entire time basically grazing on food that they find on the seabed. And the way they do this is with these wonderful jaws that they have on the underside of their bodies. So they have, let's get this going and turn the sound off. So you can see these expanding jaws opening and closing where they kind of scrape on the seabed, grab this food and then pull it into the body. And what we're seeing here are these 5 arm sort of teeth. And when we look at it from the side, this is the teeth we can see extending out of the body. And when we look inside the animal, behind that is this big, magnificent jaw behind it. And we call this the Aristotle's lantern. So these are very effective grazing animals, to the point where some people may consider them a nuisance species because they have been known to create problems and grazing on our valuable kelp ecosystems, especially when urchin populations can expand. And I know this has been very problematic on the west coast of the US. But what we are very excited about in terms of sea urchin production is what they contain. And this is all driven by food supplies and feeding activity. And it's this beautiful, bright orange, yellow, and big and bold edible gonad, or roe, or as the Japanese call it, uni. I've also heard it being called green gold, which I think is very fitting. So this beautiful tissue here is what people consume. You can have this raw, you can have it as a food ingredient, and we'll give you some examples of those in a moment. And what we see here is a perfect example of what we're looking for in the market, these wonderful marketable characteristics. Firm roe, nice colors. We have these 5 pieces, these segments in here. And the size is very important as well. So the way that we measure size of this edible tissue is that we can weigh the gonad and then express that as a proportion of the whole body mass, and then express that as a percentage. And we call this the gonad index. And ideally, what the market is looking for is for a gonad index between 12 to 25%. And what we see here is probably around like 20%, I would say, or 22%. So nice big size. And obviously taste is very important too. Perhaps you can kind of add into the chat so we can look at this later, whether you've tried sea urchin and whether you like it. I'd love to hear your opinions on that, or you can email me. I'd be very happy to hear your opinions on this. But for those of you who have never tried sea urchin before, if you imagine what a clean ocean smells like and translate that to taste, then that is what a sea urchin tastes like. So I dare you to go out and try this wonderful animal. It's seasonally available, and it's available now. We can harvest these in the Gulf of Maine from about October, November time through to about March, April. Anything outside of that time is when they're in their reproductive season, which means when we open them up, they tend to shed their eggs and sperm, and they quickly decline in size and darken in color, meaning they're no longer suitable for the market. So again, a seasonal market. And the way they're kind of getting processed is here we can see in the processes, you can see all these urchins on these benches here getting shucked by many of these harvesting staff workers here. And all they're doing is trying to collate these wonderful edible tissues. So here we have a video showing this process. We have these specialized cutting tools like we would have for oysters, for example. And then you can see them scoop out this wonderful green gold onto these mesh baskets. And you can see it's nice and firm. It keeps its shape as they're scooped out, so they're nice and firm. And then they're kind of washed in this kind of briny solution to kind of get rid of the excess sort of residual tissues. And then what we're left with is this lovely, beautiful, fairly consistent size and shape gonad lobe or uni, pieces of uni. And then they kind of processed onto these kind of like uni trays that you can see here. So in this kind of industry, we're kind of looking for something, the processors may be looking for a product which is pretty consistent in its shape and form, especially for the sushi industry, because here we have this lovely consistent shape. And they're looking for like a fairly consistent size as well to suit the sushi industry. So we're looking at grade A sushi uni here. And we can buy this online and from retailers. These are coming from 2 processors that are currently active in Portland and Maine. So here we have some tubs from Sopo, and we also have the Brown Trading Company. We have this bamboo tray. And in general, we're looking at about $40 to $50 per 100-gram tray of these segments. And you're probably, for each of these trays, you're probably getting about segments from about 4 to 6 animals. So it can be quite high value, which is probably no surprise that there's an active fishery right now. And something we do recommend is direct marketing. So you can kind of sell whole animals. You may be able to kind of fetch better prices depending on the markets that you intercept. But in this example here, you can get you can buy a green sea urchins whole for about $30 a pound. And each pound, depending on the size of the animals, you're probably going to get about 3 to 5 animals. And they're very versatile in terms of how you can consume them. So here we have a couple of lovely examples where not only can you have them raw as sushi, you can have them as wonderful ingredients in meals. You can kind of use the uni to create sauces for pasta dishes or noodle dishes, for example. And here is an example of a dish that I had from a tuna oyster bar based in Rhode Island a couple of years ago, where we had this wonderful dish of green sea urchin on the seabed. Seaweed bed there, which was absolutely delicious. So it's very versatile for this product. And if you're wondering about how to use uni, a lot of these sales, these retailers online actually have a lot of great recipes you can find and use. So if you're looking for inspiration, do take a look online. There's some really great examples there. So at the moment, green sea urchins are largely produced from an active fishery. They used to be harvested across Maine down to Massachusetts, but because of declining stocks, we are seeing this activity solely happening in Maine now. We do see other activities across the Canadian coastline as well. These animals are subarctic species, so you can find them off Alaska as well and Norway. But here on the East Coast, it's just primarily active in Maine, although urchins can be found present in cold Atlantic waters down to Massachusetts and near Block Island, but they're most abundant up north. And like any kind of industry, so here we have the sea urchins collected by a fisher, and there's different ways that we can collect these by hand, by rake, by trappers, by dragging. And this fishery is enforced through the Department of Marine Resources in Maine. And this is done through a process which is informed by industry. So you have harvesters, processors, and scientists working with DMR to kind of decide and scientifically inform management. And those regulations are very adaptive to help sustain the stocks that we do have. And these include gear restrictions, catch size limitations. So at the moment, the urchin size that you can legally catch as a fisher that's licensed is 53 to 76 millimeters. And we have legal harvesting periods, which are about 15 to 40 days across 2 kind of active zones across the Gulf of Maine. But like many kind of industries where we have a natural exploitation, we have unfortunately seen a decline in the fishery stock over time. So in this figure, what we can see is the landings, and these grey bars are the landings of the green sea urchin. So on the bottom here, we have the year or decade. And you can see in the late 80s to 90s, we had the peak of the industry for where we were landing about 35 to 40 million pounds of green sea urchins. And this was equivalent to about 30 to 35 million dollars. And this is on a per annual basis. So this really illustrates this really can be a very lucrative industry when there's sufficient stock. And unfortunately, despite the great efforts of DMR and increasing regulation, stocks have declined. They have kind of stabilized. In some areas, we're seeing a little bit of expansion, but we don't really see the fishery as having any much room for growth at the moment. So what this ultimately means is that we have this unmet demand. People want uni. We need domestic supply. There's also, supply demand internationally for this product. And we have a significant economic opportunity here. Because of the declining stocks, or limited stocks, I will say, processors are at times having to source from competing areas like Canada and Chile, but they do want domestic supply. So what this really highlights is that there's a significant economic opportunity here for the area of aquaculture. So this is something that I have been working on with a great team across Maine, including Dana, who's joining us today, where we've been looking at expanding the emerging sector of aquaculture to help sustain this industry and take advantage of this this really exciting opportunity. We have everything we kind of need to allow that to happen. Like the oyster industry, where we have these different components to support aquaculture, we do have an active hatchery. The region's only operational hatchery for now is based in the University of Maine Center for Cooperative Aquaculture Research, which is just right next door to Ellsworth in Maine. And here you can see pictured Steve Eddy, who's the director of that facility. And just for reference sake, I'm going to call this hatchery the CCAR. They use the acronym CCAR hatchery. Otherwise, it feels like it's a bit of a tongue twister. But the CCAR hatchery is the only operational seed supply at the moment. And the way that CCAR operate is that they are using this to help support the industry in the hope that over time we'll see a commercial expansion of hatcheries so that they can help kickstart that process. And looking at the kind of production cycle, you can collect the adults from the wild. These will be your brood stock. You can use these to feed them up, to fatten them up, ready for spawning or reproduction. We can go through these wonderful exotic kind of looking plankton feeding larval stages that takes about 3 or 4 weeks until the animals are ready to settle and transform into these juveniles. And then, so that whole process will take about 3 or 4 weeks. And then when the animals are settled, we can look at another sort of 6 to 12 months where we get those animals to start feeding onto more complex diets like kelp and get them large enough so that they can then be deployed to farms where we can have growers that may have shellfish or maybe growing algae and looking at interested in growing sea urchins as well. So we can deploy those seed to the farms. And then it will take about 2 or 3 years at least for them to reach market size of above 50 millimeters in size at those farms. So it's a slow growing species, but there's some benefits to this. So one of them is that we know that this animal can be very good at coping with things like climate change, especially ocean acidification, where we're seeing changes in seawater chemistry. There's been studies that have shown that the animals are very good at adjusting their physiology and they can continue to reproduce. So we're kind of looking at this as a tool for not for monoculture, but polyculture, where growers who may be looking for diversification of what they're producing in their farms, the contingency against losses that we face with climate change, this could be a really great tool for that. Given their high value, this could be an interesting option for those growers. So in the hatchery, they follow kind of general practices like the ones that I have done for several species for Europe, across Europe. And here we have an example of the brood stock, those adults, where we have them in these tanks in their hatchery and we fatten them up with lots of kelp. so that we can get plenty of eggs and sperm out of them needed for the hatchery. And there's a few ways you can induce the animals to reproduce. You can use like heat shock like we do for oysters, but in the sea urchin world, we tend to inject them with a strong salt so that they then reproduce or spawn. And here I've got a couple of videos. So here we've got a female who is basically pushing out eggs from the gonad through a small pore above the gonad. So you can see that spraying out there, and you can see all these beautiful bright orange eggs falling to the bottom of the jar. So having this kind of isolation in this jar is really helpful for us because it means we can kind of collect these eggs and control when and who is being combined with who. So we can really understand which fathers and mothers and fathers are going to be partnered together for that hatchery. And you can see that they're producing a lot. These animals can produce hundreds of thousands, if not millions of eggs here. And I have on the right-hand side a time lapse of a male. So you can see the sperm coming out from each of those 5 pores above the 5 segments of the gonad tissue. And this is actually a different species on the right-hand side. This is a purple urchin, which is native to Rhode Island, Arbesia punctulata. And the reason I'm showing this is just to show that this is a versatile tool that can be applied to many different species, and we can induce both these species to spawn. So we have them release their eggs and sperm. Then we can combine those eggs and sperm into a bucket containing clean, filtered, UV sterilized seawater. And then they can go through fertilization, which you can see in this image here with the fertilization membrane. And then they go through cell division, which you can see through these different pictures, until they then become more complex embryos and start to resemble a miniature larva. So here, after about 2 or 3 days, you can see that the gut is starting to develop in this embryo. And then here we have this kind of basic prism shape emerging. We have a fully established stomach, and the gut and mouth are just being finalized in their development. So what this means is, we can then transition in this high density holding conditions that we have in these buckets and now transition them to larval holding tanks. So here we have a Seacar Hatcheries wonderful hatchery manager, Liz Cogson. And she's been doing some amazing work at this hatchery and helping us with a bunch of projects where we've been working together to try and optimize hatchery production using our pooled expertise. And I'll talk about some of those challenges we've been addressing in a moment. But here, these tanks, you can see these are huge 250-litre tanks. These are conical tank designs, so they kind of have a inverted, they have like a V at the bottom underneath the bench here. And you can use these tanks for like a partial slow flow through if you have an inline filter to keep the larvae in the tanks. But I think what most people tend to use is a static water design where they'll not have a water flow, they'll just have water in the tank that's clean and highly filtered. And then when they're ready to kind of change the water, they can then collect those larvae in a filter, exchange the water, and then put the larvae back into the water of those tanks. And that seems to work quite well for different hatcheries. This is me setting up some time ago now in the Scottish Association for Marine Science, a hatchery for European species that I worked with, but it's just to highlight that you can use these flat bottom bins as well, using a static water exchange design. And in any of these tanks, you can stock these larvae at about 4 or 5 larvae per milliliter. So you can get some pretty high stocking densities there. I apologize about that. There we go. And I actually prefer these bin designs purely because they're stackable, so they're a bit more space friendly. So you can use these different kinds of approaches. As long as they've got some aeration to keep those larvae suspended and the microalgae food items suspended, then you're in a really great position. So here's some larval pictures up close and personal, so you can kind of get an eye for how strange and wonderful looking these things are. So here we have a 2-day-old larvae. So you can see the stomach here. We can just see a bit of the gut. And what really distinguishes the kind of different development stages in these larvae is the number of arms that are present on their body. So on this larvae here, I'm hoping you can see my mouse move. We can see some basic, a pair of arms here appearing like little stumps, and then the 2 more at the back there. So this is a 4-arm stage. And this will dictate how much food we give it. And it's usually some sort of number of set of cells, and everyone has a different approach to this. We would typically feed these animals maybe about 1,500 algal cells per milliliter per day, and that'll be on a per larval basis. And you can just see the line here. This is its skeleton that's forming as well. So they have these really fine skeletons inside. And then later on, we're going to zip ahead. They grow an extra pair of arms, so they have a 6-arm stage. And unfortunately, I don't have a picture of that here. But the next stage is an 8-arm stage. So here is this picture. So we have one pair of arms here. We have another pair over here. And then the third pair are on the sides. And then the fourth pair are just appearing underneath here. So again, this is a good visual cue to indicate when the next sort of like increase in food supplies need to be. And at this point, I would probably feed these larvae 7,500 cells per milliliter per larvae per day. And then when they get ready for competence for settlement, we can use another indicator here. So this is the kind of stomach, nice and big here. And right next to it is this site that's called the rudiment. And this rudiment will start to see a tube foot extend from this site. And what that tube foot is going to do is try and detect a suitable substrate to attach to. And then when the sea urchin is attached to it, it'll then transform into this newly settled juvenile. So we use that as a great tool to kind of figure out when to put them into the settlement tanks. And here is just another example of just showing some features of these larvae. So this is this larvae's got 8 arms or 4 pairs of arms. We've got 1 pair here, 1 pair here, another pair here, and the 4th one's just appearing here. This is the mouth. Then we have the gut in this section here, and this is the stomach. And you can just about see the anus just behind there. So anyway, there we go. There is your wonderful exotic sea urchin larva. And of course, like all plankton feeding larvae, we have to put in a huge amount of effort to feed these things. So there's a lot of effort in advance of these production cycles to produce vast amounts of algae to support their growth. Different hatcheries have very different techniques for this. We would probably feed them a combination of Denaliella, Phyodactylum, Catassaros. You can use these kind of, excuse me, sorry about this. You can use these kind of carboys and then use large bag cultures as we start bulking up. And I know that during our last project together with CCAR, they really sort of invested in using these industrial plankton reactors to kind of better automate some of that larval production as a tool towards optimizing methods there. So I know that these are definitely growing in popularity. So recent activities that we've been doing is partnering up with CCAR to see if we can find ways to optimize production to make seed supplies more consistent and reliable. And to do that, we kind of we did a strong knowledge exchange approach where we learned a lot about the CCAR applications, simulated those in what we can see here as a miniaturized kind of hatchery we developed in URI. And this was funded by the Northeastern Regional Aquaculture Center. And the person you can see pictured here is Tara Plee, who recently successfully defended her PhD and is now working for the Department of Environmental Management. But Tara was great in helping us sort of lead the way of like trying to optimize those methods by looking at larval production. So here we'll explain a bit more about what we did. So when I moved here a few years ago from the UK, I immediately reached out to Steve Eddy at the Hatchery to kind of like learn more about the industry and identify whether there are any challenges and to see if we could like pull our expertise together to sort of find ways we can try and help use our expertise to help the industry grow. And one of the things that Steve had mentioned to me is that at that time, the settlement success of those competent larvae and post-settlement survival was very low. It was like as low as 1 to 5% and was a bit of a bottleneck for them. So they were really looking for ways to try and enhance that bottleneck there. And the way this is like typically looks with sea urchin hatcheries is using this kind of high surface area wavy polycarbonate plate system. So this is a picture I took from a commercial hatchery that used to run in Southern Ireland. Where they would use a hatchery to seed a local sea loch to supplement the fishery. And what we do with these plates is we create intentionally this biofilm. So you can see this fuzziness in it. You can see all the dots in here. This is Paracentrotus lividus, so it works very well. But the biofilm is really important because one, it tells the sea urchins that it's a good place to settle onto. So it acts as like a cue for them to settle onto these surfaces. And two, it acts as a very important food source for those newly settled juveniles. So these juveniles, because they're so small, they have such delicate these delicate teeth, those Aristotle lanterns I showed you before. They can only use these kind of basic diets, these soft kind of biofilm type diets. And then as they start growing as a juvenile, they can then start accessing more complex diets like macroalgae. So it's a really critical point of the production process to make sure we can find the optimal sort of settlement media and post-settlement food supplies. And every hatchery has an approach to doing it. It doesn't mean it necessarily translates well from one hatchery to the next, purely because water quality conditions are different, the biological activity in those areas are different, maybe the population of the urchins' preferences may be different. So it's very difficult to translate all those results. So it does sometimes mean we have to kind of do large scale experimentation to try and figure out, what's the best approach to resolve that. So this is why I really enjoyed this kind of partnership project, working with Sika and also with Dana, who's going to speak to you in a moment, where we did this strong knowledge exchange experiment, like I mentioned before, where we can use university spaces who have the luxury of space and resources to do large scale experimentation, but industry, a bit like CCARV, really perhaps can't afford that or don't have access to that. So I think it's a really great way to kind of partner like people together to utilize those resources. So Tara led this project where Tara looked at a range of different things, looking at different biofilms. So looking at like locally sourced and isolated biofilms. Even looking at whether the presence of sea urchins could encourage and create a chemical cue to assure these juveniles that it's a good place to settle, indicating there's a population there, for example. And then Tara also pre-treated the seawater with like known food or habitat macroalgae cues, using adults as well that had been sat in the seawater, looking at different temperatures, and also tried different feed regimes and all sorts. So it really was a large-scale experiment there. And Tara did a fantastic job in getting this together, and it's published. This work is now published in Aquaculture, so you can access that in the QR code here. But some of the kind of key results from this was that Tara was able to improve settlement success by up to about 80 to 90% by creating a biofilm in the presence of adult sea urchins, which we thought was really interesting. And she also showed that providing ulva, this like sea lettuce macroalgae that you can get sourced locally from the coastline, also promoted the best growth for those young sort of juveniles. So those results, those best performing results, are now being kind of trialed in CCAR at an industrial scale. And while I don't have the actual number of what that looks like now in terms of the post-settlement success and post-settlement survival, I do know that this project, along with other projects that we've had along the pipeline, have helped kind of to enhance production at the hatchery. So we're really excited that there's been some positive outcomes of that partnership. So we had these animals, we had them on these settlement plates, and we then encouraged them to grow and then transition them onto more complex diets. And then we can put them into these kind of these nursery phase tanks to get them up to the right size so that they can be implemented to the farms for grow outs to the market. And this can be done in a different a range of tanks. So in this picture here, you can see my own line manager from Scotland. This is Maeve Kelly, and this is my one of my collaborators, Rachel Simmons, from the University of Liverpool. And this was part of an EU consortium grant that I was part of, where we were trying to optimize sea urchin production in Europe. And this picture is this purple sea urchin hatchery in Ireland. And you can see they had such an incredible facility here. They had these stacked kind of cone-shaped tanks where they kept these juveniles, which you can see in these dots here, and they would feed them macroalgae until they were big enough to deploy into the local sea loch there. But we can use a range of different tanks. We can use regular tanks where we can maybe hold the urchins in these baskets. and the urchins will occupy any kind of benthic space that's available there, and then grow those until they're ready for deployment. So when it comes to deploying the seed to the farms, we ideally want them to be about 10 to 15 millimeters in size. So it's probably about right for what we can see here. And I want to thank Tara for allowing me to use this picture. And the reason that size is great is because one, it's they're large enough to be kept inside the mesh sizes of general aquaculture gear, which is obviously very important. But also because at this size, we know that the urchins have a very high survival rate of being transitioned to the kind of more variable and natural environment than what they're perhaps used to in the tanks in the hatchery. So there's been a lot of research that goes behind that. So when we deploy them to farms, well, how should we grow them? Well, we know that sea urchins can be grown in a wide range of gear, which makes them very versatile. But what we do recommend is that that if you are considering growing green sea urchins, is that you consider on-bottom gear. So you can use these oyster bags and these cages. You can also potentially use like converted lobster pot cages as well that can go access to the seabed. We also recommend suspended gear that goes to sort of like submerges the animals to kind of mid-water levels. So you can use things like lantern nets or these kind of oyster baskets. And the reason we recommend this is because sea urchins really need to have access to cool water. They're a cold water species. So if you have a site and your water is below 19 or 18 degrees Celsius, which is about 65 Fahrenheit, and if you have a salinity of about 32 to 36 parts per thousand, maybe salinity 30, and you have medium to high flow rate, then you may have a site that could be suitable to hold sea urchins. So using these kind of gear can work really well. Okay, so that's the holding conditions, but what about space or scale? So this is something we're still kind of getting some details about, but sea urchins grow attached to surfaces. Spine length often will determine the space they can be grown in. So it's best to assume about a 1-inch space around each urchin. And if you're thinking about different scales, if you're thinking about doing this at a hobbyist scale, maybe less than 5,000 urchins, yes, they can be easy to grow, but it's probably not going to be very profitable. In an intermediate scale of like 5,000 to 50,000 urchins, could it be profitable? Maybe, we're not sure. We're trying to figure out the scale to operate at, but a lot of that is dictated by effort on the farm, and we're still trying to get a lot of information on that from growers at the moment. But we're very fortunate to have some really great growers that we work with who are helping us learn more about that. But if we base this on the methods that are used in China, let's just say we have a harvest density of about 50 to 80 animals, which are about 2 inches in size. And we have this on a per square meter or per 5 to 9 square foot. And let's just say in theory, you have 25,000 market size urchins. They will require about 2,800 square foot surface area. So what is this equivalent to gear? Well, if you have cages at the bottom of your farm, and they're by, let's just say 2 by 3 by 4 foot, that means the space available for the urchins to occupy which includes the bottom and the sides of that gear, equates to about 40 square foot, which means you'll probably need around 70 cages to support 25,000 urchins. And that's obviously thinking about the end point here when you get them to the market size. That will be less as they occupy small when they're smaller and require less space. So anyway, just to kind of give you some sense on that. I'm happy to address any questions on that later. And of course, what about feeding strategies? Because that's going to be a really important part of this. So with urchins, we know that they're excellent grazers. So they can and do graze on detritus, biofilms, and biofouling on the gear. And you could, in theory, rely on that alone as a low effort farming effort. But it does mean that you're going to have probably a low gonad index, and it's going to take a long time for them to get to the market. You're looking probably at more than 3 years to reach the market. So what we recommend is a medium effort where you supplement this with algae, which you can see pictured here. And I thank Stuart Hunt from Casco Bay Farm, who grows urchin, seaweed, and scallops at his site, and we'll talk about that more in a moment. But here you can see he stuffed a bag that contains hundreds of urchins with this algae. They like sugar kelp, and we know that's something that's produced by many growers. They like alaria, dulse, sea lettuce. You can give it to them fresh, dried. And I'd recommend providing this at about maybe 2 to 4 times a month. And what this will do is will give you a much more higher gonad index, making them much more marketable. And you can reach some of your products within 2 to 3 years ready for the market. So how the cost associated with this effort will vary depending on how much effort you want to put with the feeding strategy that you have, whether you want to aggressively feed them, maybe feed them less aggressively, or not feed them at all. But we definitely, for the best results, it's recommended that you do provide some seaweed if you can. I wouldn't recommend using rockweed or fucus. My understanding is these algae have chemical defense, and that is something that isn't appealing to the urchins at all. Okay, so that is feeding strategy. So thinking about that, there could be some interesting opportunities here. If you're growing sea urchins for several years at your farm, this opens up avenues for those who are seaweed farming, where potentially, if you can grow seaweed outside of the traditional seaweed growing season, there could be an interesting benefit for more than one industry there. So just for some food for thought there, and the pun was totally intentional. Okay, so I'm about to hand over to Dana, so you can stop listening to my boring British voice. But Dana's going to talk about one of the key challenges that we've been working on through the Northeastern Regional Aquaculture Centre funding was what Steve Eddy had suggested to me when I first got here was that uptake of the green sea urchin aquaculture sector had been pretty slow. And it seems that maybe because people weren't even aware that green sea urchin aquaculture was an option, perhaps weren't aware that seed were available, maybe they didn't have experience in this. So I was really excited to partner with Dana and Steve and Luz, and I think Larry Harris was part of this project as well, and some really great growers to try and address this problem by increasing awareness and uptake. So I'm going to hand over to Dana now, who's going to talk about this. Thanks, Dana.
All right. Hi, everybody. And thanks. Yeah, the phenomenon of trying to grow oysters has been an interesting one to be part of and to watch because there's been a fair amount of effort over years to try to make this as a profitable species to grow. But as you can tell, you start to get the sense of some of the challenges in there. And one of the big challenges, and I think to Coleen's credit, is you got to start with some babies. And we found this when beginning with scallop culture, that whole industry, if you will, started with spat collection because the whole cycle, either for stock enhancement or for intensive culture, starts with a seed source. And urchins have those challenges. And so seed sources have been in short supply and kind of sporadic over the years. And one of the really nice things about the work of this group is that having seed to be able to share with industry people and to be able to put those connections between the science expertise and the industry expertise, just the simple fact of being able to put urchins out and do some experiments with them and observe them and kind of play around with them a little bit has opened up all kinds of opportunities for more knowledge. And that's one of the reasons why I really enjoy my job is because I get to work with the industry side of the expertise as well as the academic and scientific. And that's really where the stuff really starts to happen because you need a lot of experience, you need a lot of brains and eyeballs and hands on a problem like this. And so having accomplished that pretty difficult job of supplying seed and starting some of those grow out trials, that meant that part of the job was to tell people about this. And here's a short list of some things that we have done over the years. There have been some press releases and some stories about the work that has happened. We have someplace in the basement of YouTube recordings in our accounts. We have a recording of a workshop that we did a while ago. We've done a number of presentations, say at the NACE conference and other conferences about this. We do have an email interest list, which does not get a lot of traffic, but that's kind of by intent. And it's a way to network people together, to share research results, to ask questions so that we can all kind of move down the road together. And in recent years, through the grant funding that Coleen's been able to acquire, that's allowed her and the team to take some of these seed and pass them on to producers who then basically say, hey, if you give me some free seed, I'm game to talk with you and show you what I'm doing and share some data and all that kind of stuff. So that's really opened up some doors. Next slide, please. And I can't remember quite how many slides there are, so I'll just kind of keep going until I get the high sign. Um, so in short, and this looks like a pretty simple slide. It's not a slide that's covered with dots or a map that's covered with dots, but don't let that fool you. So we have growers who have been working with urchins for some years, and then through the access to the seed, that's brought a new suite of growers together. And that has, as I say, opened up some real doors and made things productive and exciting and avenues to really put pieces together. So we've got more growers around the coast who are experimenting with urchins. And when I think back to what's happened with scallops, the very same thing happened. It was an interested, committed, passionate group of people who were willing to put the time and energy into trying something completely new with no guaranteed outcome. But because they were interested and energetic and observant, that mix of observation and scientific expertise really helped to move that species along. And essentially, the same kind of thing is happening here. There is a fair bit of interest. And again, seed supply is still currently an issue. There seems to be pretty good potential for private seed available soon. And I think we've all got our fingers crossed for that because that could continue this pathway to get product into people's hands and to let them experiment with it. And here's a quick slide. We do have some resources, and I'll see if I can gin up the recording of that workshop that we did. The email list, you can drop Coleen or me a line in case you'd like to join that. Would love to hear from you. Luz Cogson, who's pretty wonderful. She's up at the Seacar facility. If you're interested in getting some urchin seed, she's the one to contact. And Colleen, you have to remind me. Oh, there we are. The QR code. So Steve Eddy led us in developing this document, which is 12 or 14 pages or so. It's a pretty good coverage of the basics of production, some of the market. And it's a good free resource. So I encourage you to either use that QR code or go to the URL and download it.
Okay. Do you want to say anything else, Dana, before we kind of transition onto this?
I will. This is the part where I'll probably jump in. I don't mean to
Sure.
interrupt, but I'll either raise my hand or give you the high sign or something like that if I've got something to add.
That's great. I like that. You know, it feel more like a discussion. So I'm happy with that. That's wonderful. Well, thanks for that overview. And, to say, mentioned earlier, Dana really has been instrumental in helping us reach out to those different communities of people to encourage uptake and promote awareness. I know that Sea Grant extension staff really have this wonderful trust that they've built with these communities. So it really was fantastic to be able to see that in action. So thank you for those wonderful efforts. And also, I'm sorry my camera wasn't working before. Now it's functioning. So now you can see my face. I do not look like a sea urchin as far as I know, but here we go. So yes, so a lot of that, a lot of what we've spoken about is looking at sea urchins in the form of polyculture, where you can dedicate space to hold urchins in dedicated gear where you may have, several species growing in your permitted space. So you may have like, I know, scallops, algae, sea urchins, but some work that I got involved with when I was working on the west coast of Scotland many years ago, working with urchins, was that we were kind of starting to look at integrating sea urchins and using their grazing feeding habit as a way to try and intercept and reduce problematic biofouling that I know the shellfish industry faces issues with globally. So for those of you who have joined us today, if you're not familiar with this process, I'll just explain that briefly. So, from spring to fall, our water is incredibly productive. There's a lot of biological activity going on there. And we have all these like really interesting like critters who are reproducing and trying to establish themselves across the ecosystem. And what that ultimately means is across the season, we'll see a lot of settlement of these young juvenile mussels, barnacles, sea squirts, and they tend to cover gear. And you can see one picture here that was, I can't remember if it was from Dana or from Andrew Peters in Vegetable Bay, but it's just an example here of, the heavy biofouling that we can see on gear. And this is problematic for a couple of key reasons. One, it adds tremendous mass to your gear, making it very difficult to handle. So it means we have to kind of like deal with that and remove it, or we may need to change the gear. And then, as we put clean gear in, take away the old gear and clean that out. So there's a lot of effort required. And that also requires, access to, well, having to maybe transport teams. There's a lot of investment and effort behind that. But also it can kind of reduce the amount of water that could be flowing through your mesh of your gear. And that's really important because, and that's a serious problem because many of these shellfish really depend on having lots of water flow. So it brings all this wonderful organic food supplies to them that they get from the water. So we really have to handle this biofouling. So growers, a partner grower like Stuart Hunt, who runs Casco Bay Mooring Farm up in Maine, he was saying that he uses lantern nets, which we can see here, to grow his sea scallops and on-bottom cages that we see pictured here. And he's starting to grow sea urchins along with kelp. And I think Stuart was saying that he probably has to change his lantern nets maybe at least 3 times a year to kind of handle the biofouling. And I know this is a constant practice for, larger scale farms as well, like oyster producers, for example. So if we can bring in urchins and have them cohabit the same space, so in this example here, if we can maybe integrate urchins with the scallops in this compartment, in this lantern net, maybe the grazing activity of the urchins could be used to reduce that problematic biofouling and maybe reduce the amount of effort that's associated with that, maintaining and trying to intervene with that biofouling. So I'm very fortunate to have a wonderful research associate, PhD student, Brendan Elber in my team, who came in with some really lovely aquaculture experience. And he's also a Sea Grant fellow. I know he's very busy getting ready for fieldwork, so isn't speaking today, but he's been leading the charge for this really interesting range of studies where Brendan is trying to establish the kind of ideal stocking densities of both the scallops and the urchins and their size classes to try and find that sweet spot for integration with these species. And the reason the size classes is important because you cannot really place really small shellfish with very large urchins because the urchins could graze on them. So as long as those shellfish are large enough, then the urchins can then graze on the surface of those shellfish to remove biofouling off their shells and the gear without any negative effects on those aquaculture species. So we're trying to find that kind of sweet spot there. So Brendan's really busy processing a lot of this data. He did share this wonderful picture with me, and I hope he's okay with me sharing this, where the last experiment that Brendan finished, he shared this picture with me, and I'm going to get the audience in a moment to prepare yourselves to put something in the chat. So we want to know from you whether you think our experiment worked of integration. So in this lantern net, we have 5 compartments. We have compartment number 1, compartment number 2, number 3 here, number 4, and then number 5. So I'm interested, and in here, we have some compartments that contain sea urchins. All of them contain scallops, but some compartments do not contain sea urchins. So maybe you can just add into the chat quickly which compartments you think contained sea urchins just by looking at this image and based on what I just told you. So I'll give you a minute or two, but I know Meredith is going to help me pull out a couple of examples from the chat. So do we think this experiment worked? Oh, I don't know. Drum roll. So I'm hoping we're going to get some responses. Oh, I can see some activity. Oh, this is exciting.
Yeah, they're coming in. And yeah.
Wonderful. Okay, so what can you see a couple of examples you can read out, Meredith?
So it seems like in general, your question was which compartments have sea urchins, and a
Yeah.
lot of 1, 3, and 4s and 1 and 3s.
Okay.
I
Yeah.
also just made I mean, I just said it differently. I was like, number 2 seems like it could use sea urchins. So yeah, a lot of 1s and 3s and some 4s.
Okay, that's great. Well, thank you so much for being
Yeah.
such an engaging audience, and thank you, Meredith.
Yep.
So yeah, you're right. We know for sure that compartment number 2 did not contain sea urchins. So did integration work? Yes, I think so. So I'm happy to see that you're all sort of natural observers of science here. So we're obviously amongst a really great community here. And again, some like great photos that Brendan kindly shared is internal shots of those compartments of the net that you just saw. So on the left-hand side here is the compartment that had no sea urchins. So we consider this a control because this is telling us how much biofouling there would be normally without the urchins. And you can see it's pretty gross in there. There are a lot of these sea squirts in here. You can see there's a lot of debris. There's not much opening of the meshes of the net here. So Stuart in this situation would have definitely have switched this net out in this situation by now. But on the right-hand side, you can see one of these compartments that had a bunch of sea urchins. So you can see those here. And you can see all this wonderful open mesh here. So I'm really excited for Brendan because it's really shown a really positive outcome from his really interesting research. We're still trying to tease apart whether, how this partnership of these animals in this same space works. Is it having a negative effect on the quality of the oysters or not? We're not sure. Preliminary data indicates maybe not, because survival looks pretty good for everyone. We didn't see any strong negative kind of aesthetics of anything. And from the previous study we've done before, it seems like that maybe the presence of urchins could be helping some of the scallops grow enhance the gonad development in these scallops. So, for some consumers, that may be an appealing option for the market. So, but that's very speculative at this point. We're trying to get some stronger data together for that. But we're excited. I think this is a really great result. And I really can't wait for Brendan to kind of dig deeper into that to see what the big outcome of that. But I'm happy to show, some really strong impact of his research.
Yeah. Could I jump in for a quick sec?
Oh, please. Yeah, go ahead.
Yeah, thanks. In the previous slide there, I'm probably preaching to the choir here, but any of you who have raised anything in nets before are looking at the the fouled nets and thinking, okay, that's a lot of work. So all this transfers into time, which equals money. And it's kind of fortunate that some of the earlier work done out of the Damien Brady's lab with Struan Coleman and then Chris Noren um, created a thing called a bioeconomic model for scallop production, in which you can put in inputs like capital expenditure and labor. And so if you're growing something like scallops and you put urchins in there, if you're able to quantify the degree to which you're not needing to touch your gear, the labor saving, well, that has a definite quantifiable time and that has a quantifiable value. So here's a case where there were two distinct projects, but they've overlapped nicely. And so hopefully not only can the urchins do some of your work for you, but you can manage to put it on paper and look at it in a much more analytical way, beyond just seeing, oh yeah, this lantern net looks a little bit cleaner. So I've been trying to put my hands on where that bioeconomic model lives online. I can't do it, but if you're interested, shoot me a line. And in the meantime, I will put a link to that paper in the chat. Sorry, thanks.
Thanks, Dana. That's great. And actually, I forgot to say, Brendan had said to me he'd chatted with Stuart and, reflecting on the results that we see here, Stuart had indicated that if his nets were in this state, he perhaps would have been changing his nets much less, which links to that kind of economic model you're talking about there. So a tremendous kind of outcome from that. And thanks for sharing that, Dana. I think it's great to hear about the broader work that's going on. I'm sorry, it feels like my mouse is going bananas here. Okay, so Brendan also doing a tremendous job in helping us lead the way in an important part of our research as well, which is, experiential learning, skills development, workforce training, which all of us are all working towards as well to help the next generation of stakeholders, lead the way. So here, Brendan is diligently leading a great undergraduate team that we have at the moment in the lab. So we've got Karina Torres from Rhode Island College, and from URI we've got Joe Galdieri and Lila Saunders. And they're doing a tremendous job in keeping our research program going and active, using our quarantine recirculating systems. So Brendan here brought those wonderful urchins back from that experiment that you just saw from Casco Bay to our quarantine facilities, and then trained these wonderful undergraduates on how to measure these marketable features for these urchins. And they were therefore, engrossed, fully immersed in collecting real scientific data. And they did such a tremendous job with that. And this is just one example of the urchins from that farm. Now, I just want to highlight two things. One, this is a lovely specimen. And it's just a month earlier than the normal harvesting period, but it's big. We've got a really great color here. It's getting nice and firm. So I would consider this being, pretty marketable. I think you could market this no problem. But one thing to note here is that Stuart did not add any additional feed into these nets. This is solely from biofouling. So I think that's a really amazing outcome from this. And I was discussing this with Dana earlier, and we were a little bit surprised. So I think we've got some good questions to think about, trying to understand why is that, how has that happened? So I think I think Brendan's got a great task ahead of maybe trying to explain that. And Brendan and I took some of this stuff back to our homes to try. I don't know what Brendan's experience was, but I thought the urchins were great, but I am biased as usual. But they were very, very edible, which is a great outcome from that too. So, kind of thinking about the links to Rhode Island here. So, this is a production activity which is primarily occurring in the northern part of the Gulf of Maine. So where's the link to Rhode Island? Well, this is the situation here. We're kind of looking at trying to understand where are ideal habitats for green sea urchins, because even though we may not find them somewhere, doesn't mean it's not an ideal habitat, because we're trying to kind of also address the issue of limited stocks due to harvesting. And yes, there's a little bit of warming related climate change that may be occurring as well, which is also affecting green sea urchin natural food resources, things like kelp. We're kind of seeing now low grade kind of algae turf that's kind of appearing where kelp used to be. So it's a lower quality food source. So all of these kind of complex systems are kind of interacting with one another, which may be dictating where we find urchins. But I've partnered here with Andy Davies from URI, who is a predictive habitat modeler, and he's helping me kind of understand from multiple resources and data mining from literature to try and figure out where can we ideally grow green sea urchins, and does Rhode Island have a part in that potentially? We have seen green sea urchins in federal waters near Block Island. It makes sense, it's cold Atlantic waters. But at the moment, in terms of state waters, we know that Narragansett Bay and the ponds are far too warm to host green sea urchins, and we wouldn't want to put them there anyway because of the sensitive ecosystems we have. But could there be an opportunity to grow sea urchins in state waters where they're within Atlantic waters and they're cool enough? Maybe. The green sea urchins are not really considered as native in Rhode Island at the moment, despite their presence in nearby federal waters. But I think if we could find growers that have access to these cold water sites who may be interested to work with us, I'd love to have a conversation with you to kind of learn a bit more about that, and then we can discuss those with the Coastal Resource Management Council to kind of look at potential opportunities. Or even if anyone has seen a green sea urchin in cold state waters, of Rhode Island, I would love to talk to you to see if we can like try and confirm that, because this is part of the pathway of trying to figure out whether there is a an opportunity or not for green sea urchins in Rhode Island. But there's interest. The CRMC, the Coastal Resource Management Council, have allowed me to operate a green sea urchin program under quarantine. So, recirculating quarantine systems could be one avenue, but it would be expensive to kind of grow green sea urchins in that kind of operation. But it does show, that there could be interest in green sea urchin production should there be an opportunity. And this is where I think I'm kind of looking more at local resources now. We have many people in this virtual room today have probably seen this beautiful specimen here, this purple Atlantic purple sea urchin, which is native to Rhode Island state waters, much more tolerable to warmer waters. You've probably seen this while like scuba diving or snorkeling. Probably seen an aquaria. Those who are growers amongst us, you've probably seen these things on the outside of your cages. And these are just wonderful animals, and they're not commercially exploited at the moment, largely because the roe inside these animals is very small, because there isn't, maybe because of food limitation of the wild caught animals. We know that we can bulk up the roe, but the roe are very, very dark. We cannot get those roe to get to a very bright orange or yellow color. But I think, I've heard of people actually consuming these roe on a kind of personal basis. So I think there could be some potential for the food industry for these purple sea urchins if we can come up with creative strategies to try and make that appealing. Or, maybe we can use it as an ingredient in food sources like, like squid spaghetti or something, for example, where we don't need to showcase a bright color, for example. So I don't know. I'm looking to kind of work with people who work in the food industry and are very creative with these sorts of things. So I'd love to talk to you if there's any of you in the virtual room. But we do know that we can bulk up the roe of these urchins. We've done, but this is a video of a trial we did published in the Frontiers of Marine Science, where you can see it munching on a pelleted a food pellet there. I think this is a salmon pellet. And we can get these roe up to a pretty big size. So we can enhance them, but we have a few more steps to try and figure out whether we can really fully commercialize them or not. And then, but there's definitely great potential to use these animals as an integrative approach, much like the scallop example we saw before, but with oysters. And we know in Rhode Island, one of our biggest products here is the eastern oyster. And Sea Grant was very generous in allowing us an opportunity to kind of investigate this, partnering with industry. So here is an example of a bag that has a lot of fouling on it. And again, it's a similar kind of principle. A lot of effort is going into cleaning biofouling to make sure that we can grow the best oysters we can. Excuse me. And in line with that comes some challenges where social acceptance of aquaculture has been a challenging route for some growers, because people may not see aquaculture activity as an appealing visual, that activity. So if we could use like sea urchins integrated with like on bottom gear with oysters, and they can reduce biofouling, maybe that may reduce the amount of times you need to visit your farm, and maybe that will help with stronger social acceptance. I don't know, but I think these are kind of interesting questions to ask and consider when we think about some of the challenges that we face across the industry. So in this project, I know Jennifer Scapitura of Corny Siren Oysters is in the virtual room, so it's really great to see you there. I'm looking forward to catching up with you soon. But Jennifer has been an incredible partner in this project funded by Rhode Island Sea Grant, and allowing my wonderful graduate student, Christopher Jenkins, who's a master's student, to run this kind of first initial step kind of trial to see whether integration of purple urchins with oysters will work well or not. We've seen this work well in Florida, so we really wanted to see whether this would work well or not in Rhode Island. So working with Jennifer, Chris had set up a replicated design. It's a first step, so it's not a full-on production scale. We just wanted to see whether this could work as a proof of concept. So Chris was allowed to use some of the oyster bags that Jennifer has at the farm, And in each of those bags, we put 25 2.5-inch oysters sourced from the farm. And then 3 of those bags did not contain sea urchins. So that's our control. We want to see what biofouling looks like. And I just want to make it clear, we didn't do any additional activities there at the farm. We didn't do any additional like shaking or cleaning. We just left them alone to see what would happen as an initial step. And then in another 3 bags containing the same amount of oysters, Chris added 1 sea urchin sourced from the pond. And then in another 3 bags, he would add 3 urchins sourced from the pond. And the reason we use those densities is because this has worked for other species done in other studies. So we want to look at this for comparative reasons. So those urchins, they're about 50 millimeters in size. These were on-bottom bags and put into on-bottom cages, submerged in about 4-foot depth of water in Quaney Pond. And this was a limited trial study. So we did this in the late summer fall biofouling season. Excuse me. And this ran for 2 months from September to November in 2023. Okay, so what did we find? Let's see. Okay, so we've seen this picture before. This is the control, so we can see there's biofouling present there. Did our experiment work? Drum roll, wait for it. Oh, look at that. So we have some dramatic beneficial responses here where this bag on the right-hand side contained 3 urchins, and you can see immediately that there is hardly any biofouling there at all. So we were so excited about this. And yeah, I'm just absolutely thrilled. So obviously we have more steps to take, but important questions to ask are, did this have a negative effect on the oysters? Because we have to make sure, our primary product isn't compromised in any capacity. So, excuse me, sorry. Chris is kind of finalizing that data set at the moment, but at the moment what we can see is both urchin and oyster survival was very reasonable. The growth was very limited, but that's not surprising purely because it was a very short period of time, 2 months, so that's not surprising. But we didn't see any negative responses here, which is an important thing to understand. And from what I can recall from seeing the latest part of Chris's thesis, is that similarly, the oyster meat was not compromised either. It was at a similar level to control. So, no negative responses there. So I'm very excited about that. But what about the biofouling itself? Biofouling species all cause like different problems. So in this figure, we were interested to see what was happening on the actual shells of the oysters to see whether urchin presence might be helping with any problematic species. So one of these problematic species is the mudworm or Polydura. So this is this worm here which burrows into the shell, it bores into the shell. It can create these unsightly kind of mud pockets on the inside of the shell. And this can also kind of weaken the structural integrity of that. But what that ultimately means is it makes it hard to sell some of those oysters when we see these like unsightly bulges. So I know for some people this is can be problematic. So we were interested to see whether sea urchins could help reduce that. So in this figure here, we have the percentage of the oyster shells for the Polydura. And then I've used the traffic light system color code here. So the more green we see means the less we see the mudworm. So that's a better situation. So green is good. So the more green we see, the less the mudworm we see. The more amber we see, the more frequent the mudworm. And then the danger color, the more abundant we see the mudworm, the more red we see. And I'm really excited to see in this figure that when we have 3 sea urchins in that bag, where we saw the tremendous reduction in biofouling, we see hardly any mudworms there. So we're super thrilled to see this. And we saw similar kind of results for barnacles, tunicates, and algae. So very exciting outcome from that. So really grateful for the tremendous support from Rhode Island Sea Grant and Jennifer's team in helping that happen. So good initial insights there. So kind of just to wrap this up, so there's something interesting work that's going on. We can see changes in practices of sea urchin production. Because we are operating this talk from the Rhode Island Sea Grant, thinking about what does this mean for Rhode Island? Well, this is a work in progress for Rhode Island. We are looking for more growers to partner with, especially those who use on-bottom gear. We want to see if some of these results might be repeatable. I'm also really would love to connect with any growers that might be in the room today who have access to fairly consistent cool water at their site. we could have, it'd be interesting to kind of see if there could be an opportunity for green urchins that we can like discuss further. And also, if there's any kind of creative people in the food sector, I would love to speak with you. I want to hear about inspirational ways that we can use urchins and try and promote some of these industries. But ultimately, the best part of my job is working with a broad range of stakeholders to have like the best impact possible from this work and to think about, important perspectives of part of this. I can't do this alone. I need to work with industry to figure out how we can like expand and establish and help with the possibility of growing these green sea urchin and purple sea urchin production opportunities across New England, but also here in Rhode Island. Okay, so I apologize, Meredith. I know I'm slightly over, but there's many, many people to thank, a whole suite of undergraduates. People who I haven't mentioned yet is Bella Southwood and Annie Langston Knoll and Larry Harris. Everyone else I've mentioned earlier in the talk, they've all been absolutely fabulous collaborators helping us this move forward. And thanks to our funding agencies, most importantly, to allow any of this to happen, where we've had tremendous support from the United States Department of Agriculture, the USDA, Northeastern Regional Aquaculture Center, Northeast Sustainable Agriculture Research and Education, and of course, the wonderful Rhode Island Sea Grant. So I'm going to leave this slide here for a moment, if that's okay, Meredith, so that people can have some time to intercept this. And if not, we can maybe distribute an email with some links there. But otherwise, thank you so much.
Great, thank you so much. I was going to say, any of the links from here, if you can copy and paste into the chat if you want, but don't worry, everyone. I will be sure to include that in the upload after today's, when we wrap this up with the recording. So I'll be sure to have this information also available elsewhere. But in the meantime, any links you guys, either one of you have, if you just want to put that in the chat. And I guess now I'm kind of opening this up to questions, but I am gonna use my moderator privilege and ask the first question because I know there's a couple in the chat and we'll get to them. But one of the things I was thinking of from the very beginning when you were talking about processing, I think it was green sea urchins, it looked really labor intensive. And so I was wondering if that would be a potential barrier, because one of the things I remember hearing about was growing different species of seaweed. I think it was Gracilaria, where there was some challenges of kind of cleaning it up and making it marketable, and it's just labor intensive to get that kind of product market ready. So I guess my question is that a potential barrier, just kind of the the shucking and all the staff you would need for that?
I don't know. Do you have a response you want to start with, Dana? Because I know you work with oysters as well.
Yeah, the urchin processing part, if I'm getting the question correctly, the urchin processing part, yeah, is kind of skilled labor. And not everybody knows how to do that. On the other hand, it is a developable skill. I don't have a good sense of how many pounds per hour a person can process, but I would imagine that that's responsible for part of the high, sort of the retail value on that. Does that help?
Yeah, no, it just seems like it would be a lot of work. Not necessarily a bad thing, right? You can make the argument that there's producing more jobs, but also that there was some effort involved there.
Yeah.
I think that factors into the cost as well. Like I can't remember the exact cost of what like the growers get or the producers get, but that would be relayed into the processing for sure. Who gets that, pays for that, I don't know. But I think the direct marketing offers like maybe like lowers those barriers that you've mentioned, Meredith. And I think it's a really great question to ask. Like the direct marketing of whole animals that may help with that process.
Yeah, that was the second part of the question, was that does that kind of counter that step or mitigate that step, that will be on someone else's end? So it sounds like it. All right, so from the audience, and feel free, everyone, if you want to raise your hand too, you can ask your question yourself. Also, correct me if I'm reading your question wrong, but one of the first ones was, can a grower air dry to kill the squirts and have urchins survive? And what's the general time frame?
Sorry, could you repeat that question? There was a cut out there.
Yeah, sorry. Can a grower air dry to kill the squirts and have the sea urchins survive? And what's the time frame?
Oh, I'm guessing I'm translating this. Can you air dry like oysters in a
Yeah.
sense? No, not at all. Urchins need to be very much kept wet. So yeah, air drying is definitely something that a lot of people do, but you'd have to move your urchins into another gear, like clean gear, for example, and keep them submerged in the water because they wouldn't survive that a long exposure. So you can green sea urchins can be found in some areas of the intertidal where the temperature range is appropriate. And you can see urchins covered in seaweed. So I think as long as they kind of kept cool and moist, I mean, we've kept urchins alive in some like transit experiments we did for the EU consortium grant I mentioned before, where we had them wet and cold, and they were alive in the fridge for about a week. So they can cope with that, and they can internally regulate their cells and tissues, sort of like systems, their physiology quite well in that system. But the point is they have to be kept cool and wet. So long air drying, unfortunately not for urchins. But it would be cool if they could. Great
There was
question.
another interesting question on to what extent does the diet affect the taste? Because you had mentioned that biofouling was a potential feed process, and so does that impact the taste? Do we know?
Yeah. Yeah, it can do. I have there was I can't remember which species this was. I've worked with a lot of different species. There was some time ago I had a species that was eating bits of fragments of fish, and then we ate it and it did taste very fishy, but I'm not sure if it's because I accidentally cut the gut as well, releasing some of the fish. So but there was definitely a bit of a fishy texture. But yes, no, the diet can affect the taste. I think one recommendation would be, I think it's a great point to bring up here is if you're a grower, to kind of get the best outcome from the taste, one of the strategies you might want to consider is feeding algae maybe for a few weeks before you harvest them to the market to get the best taste and the best kind of color out of those urchins. So that would be a really helpful strategy.
One of the things you had mentioned about how sea urchins have this adaptability to different climate and ocean acidification, like and temperatures. Um, I guess uh, I just lost the question I wanted to ask about their adaptability. Oh, with relation to marine disease, because I know sometimes as waters are warming, um, oysters, mussels, and even certain kelps are more susceptible to different types of bacteria and viruses. Are you finding that with sea urchin species as well?
Um, Dana, do you have a response to this before I say anything?
I have to apologize because I was writing something in the chat, so I didn't get the whole question.
The impact of marine disease. So even though sea urchins are adaptable to changing, because I think the point was that they're slow growing, so they can adapt to
Hmm.
water temperature changes and salinity changes. But what do we know about the impact of bacteria or viruses that impact, other shellfish species or even kelp species? Um,
Yeah, boy, uh I'm going to be throwing this back to you, Colleen, for sure. Um, the My gut feeling without having a ton of expertise is that yes, any kind of stress to the normal condition or any kind of change to the normal condition is going to cause physiological stress, which weakens the ability of the organism. It is almost certainly not the same thing. But for example, down in the Caribbean, the urchins down there have had cycles of disease go through there. And what that's responsible for, I'm not sure. But in the general case, I'm guessing that urchins can be susceptible to a range of pathogens and parasites and things like that. So definitely something to keep an eye on. But Colleen, I'm looking at you directly for uh, expertise on
Sure.
the parasites and pathogens.
Yeah, this is actually
And
outside my expertise. I mean, this is why like partnering, getting collaborators is important because they all bring different expertise. Would sea urchins be vulnerable? Probably yes. Do I know what that is exactly? No. But what we do see in the field though is I can't remember the specific name for this, but There have been incidences where you can kind of see this kind of black kind of spot. I don't know if it's the same thing as balding disease. I'm not too sure, but it's where we see an infection gets onto the surface skin of the urchins where there may be damage. And then gradually it can degrade the quality of the animal and they will slowly die. Can they recover from this? I'm not sure. Do I know what that is exactly? No, I don't. But it's something we need to know, and it's definitely one of our knowledge gaps at the moment. So maybe I know that looking at some of the people in the room, I know some people might have some ideas about that. So we're definitely interested to use the chat to kind of discuss that. But I think that's a great question. I'm not too sure, but yes, probably.
One from the audience is, if the primary urchin market is Japan, would it make more sense to encourage the urchin growing industry on the Northwest coast?
Yeah, and they are. There's the this is another emerging sector of aquaculture. So there's been an established fishery on the West Coast, but there's some newer contenders in the sea urchin industry on the West Coast where I can't remember the exact species name, so I apologize right now. But there's been a population explosion of some sea urchin species where they've been devouring kelp. There's been a lack of absence of predators like sea stars because of sea star wasting disease. So it's creating a lot of ecosystem challenges, because of the decline of like kelp ecosystems. So there's a lot of efforts on the West Coast to use those urchins, but the problem is because of the lack of high quality food supplies like the kelp, they basically have hardly any roe. So a lot of the efforts that are going on the West Coast at the moment with respect to aquaculture is that they're harvesting these, ranching them in tank systems, and using formulated feeds or similar, depending on the operation who's doing it. These feeds to fatten up the roe. They can do that in as little as 8 to 12 weeks, and then they can send them out to the market. So I think there's local supply. I'm guessing that they probably have some international supply as well. But we can't, that's a strategy that wouldn't suit us in Maine because we have a limited stock. So, it's not a strategy that would help there. But, it's an interesting mechanism because if, for example, purposes, we have a grower who's decided to use a low effort approach to growing urchins at their farm. So you have this like low amount of the gonad index. There could be an interesting opportunity to maybe temporarily house those urchins for 8 to 12 weeks to fatten them up for the market. So there could be some other opportunities there. How that would look, I don't know. Could this be something that hatcheries do outside of the season? Maybe. But these are interesting kind of areas to explore potentially when the need arises.
And I just want to remind folks that if you wish to be unmuted, just raise your hand and you can ask your question yourself if you would like to do that. I know there's some comments about kind of why exporting to Japan's. There's some back and forth about exports to Japan and why did they stop. Actually, I'm going to kind of circle back to that, but one was about how important is the water temperature for sea urchins? Is there any way to have a controlled environment in parts of New England that don't always have kind of an under 18 degrees Celsius water temp?
Yeah, it's good. It's a good question about the temperatures. So general, like the optimal temperature for the green sea urchin is about 12 to 15 degrees Celsius. They can cope with warmer temperatures. So we know we can grow them at about like maybe 18, 19 for short periods of time, maybe a little bit higher, but it has to be for a very short period of time. But we're really trying to get the fine-tuned detail of this. So something I think is a broad industry need, not just for green urchins, is to get stronger seawater parameter characterization at farms so that we can better understand what temperature variation looks like, even what salinity variation looks like, so that we can really get a kind of granular look at about the kind of tolerance range of those animals. I know that some places that we have grown urchins in open water, we can see temperature spikes like midday, probably during slack water, where we see the temperatures going as high as like 20, 21 degrees Celsius. But that's for a very limited period of time. So they can have some very short tolerances, but overall, you want to make sure that your water is just generally cool. And below 18 degrees Celsius or lower is really kind of like the best range there as far as we know right now.
And because I always have scallops on the brain, the optimal temperatures for scallop production is really 10 to 15 C. That's really the sweet spot. And so in this case, the thermal preferences and the thermal tolerances match up between the two species.
I wanted to ask, you discussed kind of the importance of having seed stock and just access to seed. For a hatchery, what's the kind of, I guess, space that, I guess we're talking like a commercial industry, like what's the kind of space that a hatchery needs for seed production or spat production?
Good question. Someone else asked me this recently as well. It depends on the scale that you want to produce at. I would need to think about this a bit more about the scale. I think Steve Eddy would be a good person to ask about this. And I can actually follow this up. Anything I can't answer now, I can follow up, and we can distribute that to the audience members if they want. Yeah, let me get back to that. I want to think about that if that's okay.
Yeah, no worries. There's a lot of different, you
Question.
described a really good process or an interesting process from kind of how you isolate the eggs to fertilization and then kind of these feeding tanks. Yeah.
I'm sorry, continue. I'm just
Oh no, I just was like, I could see it being a process that require like I could see an argument, not an argument, but a landscape where there's a small footprint, but also a big footprint. But I guess it really just depends on the scale of production that you're looking for.
yeah. And as a qualitative answer, not so much quantitative, you need X number of square feet to produce Y number of urchins. A couple of things to keep in mind is that the algal production shouldn't be overlooked because that's to do that well is difficult and reproducibly. So there's definitely dedicated space and its own set of expertise there. And then secondly, Coleen mentioned this a little bit, but I'm also impressed by what Steve and Luz have said. And Steve says that small urchins that you can just barely see like to feed on scuzz. So that was his very technical term for it. Makes me laugh every time. Um, and there is a time between settlement and when you can actually really start to see them of a kind of an extended period where it can be several weeks, where, as far as I understand it, Luz doesn't have a good sense of setting success. So maintaining the tanks, keeping the air going, keeping the water going, maybe adding some algae in there, that kind of stuff, until some weeks later you can get out the microscope or the hand lens and look at these tiny little white dots. Um, and as a hatchery operator, that would scare the bejesus out of me because I would have done all this work and then I have this black box where I don't know what happened until such time as they finally show up. And it's totally different than a shellfish hatchery where if you have a die-off, you can observe it and you can assess your success all the way along there. So there's that segment in the middle that is not for the faint of heart. And just something to know about as there might be some potential hatchery producers in the audience there.
Just kind
Yeah,
of
and sorry, it used to scare the hell out of me when I was running hatcheries. Go ahead, Meredith. I'm
I was
sorry.
just piggybacking on the hatchery aspect because when you mentioned about wild stocks kind of declining, and this would not be for green urchins in the Rhode Island area, but could you potentially use a hatchery not only to seed kind of farm raised urchins, but would you use that potentially to reseed wild stocks if there is that
That has
something
been
that,
done.
yeah.
Yeah, so that was the case study I mentioned earlier, where in Ireland, in Bantry Bay, there was a facility run by John Chamberlain. It was called, I think it was called Demana Seafoods. It was he was one of our EU partners, and he ran this facility for decades, where they overfished the natural purple sea urchin population. And this was Paracentrotus lividus, so this is the European like marketable urchin, and honestly, I don't know how he got permission to do this, but he was able to use that facility to seed the lock, and he did. Every year he would run several production cycles, which he can in the season, which is also what we can do here with the green urchins, and he would use that to seed the lock. We don't do that now, and I'm not sure what, whether that's an option for us or not. That's a conversation that needs to be had.
Actually, it's so for one from the audience. What months would farmers, possible nursery sites want urchins from a hatchery?
Could you repeat that, please? Sorry.
Sorry if I'm cutting out. What months would a farmer, possible nursery site want urchins from a hatchery?
Good question. Let's see. So it depends when they're ready, for that size. We didn't follow a kind of typical pattern in the projects we had because CCAR very graciously held seed for as long as possible to help facilitate uptake for growers when they weren't quite ready, for example, if they were getting a permit. I know that Steve has said that it's best to deploy seed before July because otherwise, when you transition the animals from the hatchery to the warm summer waters, I think it's maybe too much of a thermal shock and the survival can be lowered. So you want to hit them before that period there. So I think anything between kind of like early winter to kind of like late spring, very early summer. That's the kind of period of time you'll probably be looking at getting the seed if they're the right size and they're available.
I had a question about the difference between like the on bottom gear versus like mid-water bags and depth. And I don't know if depth is more of a function of just water temperature, or I guess it seems like some of your recommendations were for on bottom gear, and some of those operations seem like in only just a couple of feet of water. Um, and when would, maybe some of the mid-water gear be more appropriate? Or do you have a recommendation of one gear type over another?
No, I mean, I think I'm a firm believer of like maybe trying to utilize what you have already existing at the farm. If you have that kind of gear available, and it's really kind of using that. But most importantly, if you can use that gear to intercept that cold water, so it's mostly a product of that availability. We don't recommend the flip bags at all because the surface flip bags, which a lot of the oyster industry uses, is right at the surface waters. And for people who are less familiar with these, you flip them over to kind of easily access the oysters, and then you can allow them to naturally air dry to get rid of the biofouling. It's a really great approach. It works really well. It's very visual, so maybe that may have some challenges with some parts of carrying capacity. But because of the air drying and the fact that surface waters heat up quickly with like when there's like peak summer periods of the year, it's just too warm and too dry for the urchins. So yeah, basically, if you've got the right gear and you've got the right temperature water and you can access that with your gear, then that's the kind of best situation I would say for now.
And I was going to ask if there was any pushback to kind of urchin aquaculture in the same sense that you might find with oyster or kelp or any other type. Um, but I don't know if there's any growers who are just growing urchins solely, or if urchins are really more of that polyculture framework that you were talking about as like a complementary to oysters and kelp.
Yeah. I would say for the Northeast, it's very much polyculture option, purely because it's slow growing. It's going to take them a long time for them to get to the market. And you obviously, we want to make sure that people are getting income on an annual basis. So, but once you kind of get the sea urchin bite production going, you can have products every year. But it's going to take 2 or 3 years to get to that point initially. And the scale you'd need to make a profit, it's just going to be very big. So I would recommend it as a polyculture option to kind of get the best outcome from that. And it's a great contingency against losses as well, which people seem to be quite interested in, especially up in Maine, in terms of diversifying what they're producing as a contingency of building resilience to climate change.
And I think with the purple sea urchin, you said I don't know if I interpreted this correctly, but like the gonad index is lower. So
Yeah.
it seems like the market difference would be if you were to integrate it in Rhode Island, it's more of kind of the biofouling mitigation instead of it being the product that you would sell. And or I
That's
mean,
a
and
good
also what
question. Yeah.
do they taste different? Like, is it just a different color? When you were talking about market
Yeah.
um
Yeah.
appeal, I was like, does it taste different or is it just the difference between color?
Sure. Good questions there. Yeah. So um, if we collect them from the wild, the ones that we have seen from the wild have been low in gonad index, and that sometimes indicates maybe they'd be just being accessing like low quality food supplies in the wild. So some of the reasons for doing these trials with the farms is to see whether we can use biofouling to bulk up the roe or the gonad index. We can a bit. We have shown that food trials that have been done in labs where we're giving them formulated feeds can bulk up those gonad indices as well. So we can enhance the gonad. Um, the color is the big challenging part. Like I said, it's the purple urchins in Rhode Island have a very, very dark roe. It's like extremely dark purple, brown. We can get like if you look at the traditional kind of market for green sea urchin only, where we prefer these bright orange, bright yellow color roe, we're not going to see that from these purple urchins. But we can see some colors where they may be kind of like a kind of lighter browny orangey color, which may be a kind of lower grade for your traditional market of like a quality of the roe. So what we're trying to think about here is could we maybe think about a way to sort of strategically market them as they are as dark roe. Maybe that could be an interesting kind of feature, but it depends on how you package that. I'm gonna be very clear though. I haven't tried the purple sea urchins myself. I've smelt them and they smell wonderful. I think they kind of release some sort of hormone, and there's this really lovely smell that comes from them, which I think just adds to the experience of eating them as a seafood product. But I know that I've heard that people have eaten them and thought they were fine. So I don't know. I have to obviously try it myself. So I feel really bad that I'm saying it out loud right now. But there's definitely potential and people are interested. So I think that's what's really motivating here.
All right, so the next question is, and forgive me, Renee, because I'm not familiar with this acronym or term. Has anyone grown green urchins in a flupsy? Once they've reached nursery stage? Do you guys know what a flupsy is? Okay.
Dana, do you want to explain what a flupsy is?
Yeah, floating upweller system is a nursery stage equipment for bivalves, mostly oysters and hard clams, but people use it for others as well. And nobody has used that, to my knowledge. Um, it's an interesting proposition. It's a way mostly to move water, and water flow could help, um, just in the sense of oxygen and other nutrients to feed biofouling. But urchins are not filter feeding in the way that bivalves are doing. So the effect of the water flow in a floating upweller system is a little bit different. On the other hand, if you happen to have a flupsy, and if it doesn't cost you too much to operate it, it's worth a try, I think. You'd have to pay attention to, and this might be a sticking point. You might have to feed your juveniles, and that feed in the Flupsy system might interrupt flow. So you could run into difficulties there. On the other hand, as I say, if you have a Flupsy at your disposal and you have a few seed urchins, it's always worth a trial, even if it doesn't work out.
I know that Renee, it's nice to see Renee joining the group. Thanks for that great question, Renee. Do you know if anyone's using them on the West Coast, Renee? I know that Renee's a sea urchin expert working on the West Coast, so I'm curious to know if she's heard of anyone using those systems or not. Sorry to throw a question back out there. Not yet. Yeah. Okay. There's interest. Okay. Well, if you try it out, we'd love to hear more about whether that works or not. Be interesting to see that. Thanks for answering that.
And I think the next question kind of goes back to what you talked about in China and what are the grow out methods used there?
Yeah. Um, Good question. So I should know this. I know that Steve Eddy with Nick Brown put a really great Echinoderm aquaculture book together, and they've got some great chapters that cover this. So it's just called Echinoderm aquaculture. Um, do they use a whole range of things, but I think I remember standing out one example was like lantern nets. But we've been connecting with people from Japan as well. There's been a really great business guy called Kai. I think he's on the call on this group meeting today. Who's been telling us a bit about what's happening in Japan as well for grout methods. And it sounds like from what I can remember from the discussion we had, it sounds like they're using government funded hatcheries to seed dedicated ranched areas. So it's like they have like an underwater garden wall in a sense and keep the urchins in there and then feed them algae and the urchins stay there. I think it's because there's plenty of food being supplied to them in that kind of leased area. Those urchins are kind of growing there and then being then being harvested. So those are a couple of examples of what I know, but this is, it highlights, I know that Dana has been part of a big team, and I can see Hugh's part of the call today as well, where they've really led some really great efforts to try and do technical knowledge exchange with scallops. I don't know if you want to talk a bit about that, Dana, because I think this is where we need to be with urchins at the moment to try and get some knowledge exchange for urchin technology and approaches.
Yeah, I can hardly recommend it, and I'll look to friends and colleagues on here, Hugh Copperthwaite and Kichiro Hamano, because they obviously have a ton of experience to offer as well. The bottom line is that there's nothing like seeing it and putting your hands on it, and especially if you can get scientists talking to scientists and farmers talking to farmers. And if people are willing to share, it is so important. That doesn't mean that it's going to lead to instant overnight success. It might not because things are different between our waters and species. Um, but there is just so much expertise in the rest of the planet that is not the United States, and is not the Northeast, that it's a really quick way to jumpstart a research and development and industry production program. So tech transfer can really work.
I'm kind of kicking myself for not asking more questions when I was younger and I was working with this Irish producer, because unfortunately John Chamberlain retired several years ago from that hatchery he was using to seed the sea lock, and kicking myself for not asking more questions about that. I don't think I could reach out to him now, but yeah, so and in conjunction with all of this, we're seeing loss of expertise as well, which is creating some challenges for us as well. So getting updated on this and talking to one another, especially East and West Coast. I think there's some really exciting activities going on both sides of the US. It'd be great for us to kind of all get together and talk more about, how we can share that expertise and help all our, the national urchin industry grow as well.
Great. Yeah, I've noted in the chat, but if anyone wants to unmute themselves and ask any more questions, please feel free to do so. And all the materials that you guys have spoken about and shared in your presentations, I'm happy to also post that onto Sea Grant's website and cross-link with Maine Sea Grant as well, just to make sure that the resources are spread wide and far. If that works. But I don't think I have any more questions. So unless there's another one from the audience, I just want to thank you, Dr. Suckling and Dana, for taking your time this evening to join us and share all this really interesting work and the potential for a sea urchin aquaculture industry. I mean, it's there. It's just the potential for it to grow in the Northeast seems really exciting and promising. And I'm excited to see kind of the results that come out of your student work, especially within the Rhode Island area, and see how that goes. And yes, and if I guess you guys left your email, I'll be sure to include the email in the follow-up email with the recording, if that's okay with both of you, so that I know you wanted to partner with potential growers as well as foodies who might be interested in helping bring this to market. I actually think this would be a great opportunity also to maybe do an event with like eating with the ecosystem, because they do,
Mm.
you know,
Yeah.
this is kind of like right up their alley.
Yes, we've already started having some discussions with Kate Masonry. I need to kind of follow up with that, but I agree. Yeah, I think that would be really fun, a fun and interesting and important thing to do. So,
Yeah,
and it's a
yeah,
great
that was the first thing
organization. So, yeah.
first group I thought of when you said that. I was like, oh,
Absolutely, yeah. But thank you so much for hosting us today, Meredith, and organising this to you and your team. It's been
Yes,
really
thank you.
fun, and it's been great for us to, have an opportunity to share, the work that we've been doing and highlight some of these areas of opportunity. So I thank you, and thank you to the audience as well for being great participants in our questions. So thanks.
all right. Well, everyone stay tuned. I'll send out a follow-up email with the recording and notes from today. And again, thank you, Dana and Colleen, for joining us today. And I hope everyone has a good evening.
Yeah, my pleasure. Thanks.
Okay, take care.